Okay. I'm Andrew McArthur in the Faculty of Health Sciences. So I'll start up, this photo up the front. Undergrads are a big part of our research program. A lot of what I'm going to show you today, undergraduates have had a major role in. Most of the undergraduates at my labs end up publishing. Obviously, this year is harder than most in that sense, even though we are an informatics lab. So my background's pretty varied. My PhD was in deep sea oceanography, but I've worked in government. So as a fellow at the Smithsonian, I was at a place called the Marine Biological Lab. Despite its name, uses marine organisms as model organisms. So then I worked on African parasitology, and that's a 100% private research institution. I had my own company for 10 years. I've been at Mac for 6 years. So I've been around in seeds and things. And I am director of the Biomedical Discovery and Commercialization program. So I emphasize teaching business as much as I teach biochemistry, for example. Though I will readily admit I'm the only biochemistry prof in the department that never took biochemistry. Okay, so we're going to start this with this slide to give you context. So this is my grandfather's unit in the Second World War. He's this tall guy on the left with the flag behind his head. So he was a code breaker. He broke Imperial Japan's codes, right? So to decide that. So it's a good metaphor for what we do now. So as a code breaker, you have to detect signal. So in his case, he had to use really good radios to pick up that signal. And then you have to use mathematics and other methods to decode it, figure out what it says, what it means. And then you have the really difficult decision deployment. What do you do once you have that information? So my lab does the same thing. We are a threat and surveillance lab. So our detection tools are genomics. So we work on infectious agents, particularly multi-drug resistant strains, a lot on SARS at the moment. And we use DNA sequencing technologies, which, as Dr. Schellhorn said, are getting obscenely cheap as our detection. And then in the middle, we do the decode aspect. We write algorithms, we build databases, we build tools to decode what that DNA sequence means and what that pathogen could be doing. And then we deploy. So we, around the world, public health agencies use our data and our algorithms to make decisions to work on superbugs. We license tools and software to over 30 companies, and from food safety to analytics, you name it, and surveillance. And so this is a big part of our emphasis, is that we really are outward facing. We want our stuff used. And at this point, every about 3.5 minutes, someone runs a superbug through our servers from around the world to analyze. So our major emphasis is antimicrobial resistance. So we are losing antibiotics faster than we can find new ones. So every year we lose the ability to use antibiotics through drug resistance, and no replacements come to place. And the Canadian numbers, by 2018, right now, 2 years ago, 26% of infections were drug resistance. By 2050, that's going to hit 40%. So you lose the ability to do simple things like a hip replacement. But if chemotherapy might be lost, if you can't control infection, you can't use chemotherapy because it knocks down the immune system. Basically, all of modern medicine as we know it is under threat. And by 2050, we're looking at almost 400,000 lives lost, and huge economic impact. So this is the fight that we fight, and our job is to build better data and to really perform surveillance. So if you think from a surveillance point of view, you can ask, which drug resistance genes are in Canada or the Americas or globally, how they're moving around, and which ones are dangerous and really pose a threat. So sequencing is the core, and you can do that from big benchtop sequencers in a production facility, but we also use miniaturized ones you plug into your laptop. And you start with that sequence data, and essentially most workflows compare that data to reference sequences, knowledge bases around the genes and mutations that cause AMR. And from that, you predict the resistome, the battery of genes in that infection that might cause troubles. And then you do that one isolate at a time, but when you start doing that around the world, you start to build a surveillance. And then the holy grail of this is that, can you take that raw DNA sequence and predict exactly what the bacteria is going to do in infection, and to find an Achilles heel of what drug to pick? And that leads to clinical decision making. So the kind of work on this is really the three sort of areas of modern bioinformatics. The first is a subdiscipline called biocuration, which is how you build databases, how you build reference information, how do you take the published scientific literature and convert that to algorithms. And then bioinformatics is the comparative piece. Given a new sequence from a patient or from a body of water, I want to compare it to those databases and figure out what's going on. And then the last one is analytics, really where the field's heading, which is using machine learning and artificial intelligence. And our whole goal in the last 8 years is when we started biocuration, and hardly anyone was sequencing, and now public health, all of them sequence. So we started building the bioinformatics, and now all that data is accumulating. We get enough data that we can move into analytics. So on the biocuration side, we run the globe's largest database on the molecular basis of drug resistance, the Comprehensive Antibiotic Resistance Database. So every mutation and gene and every pathogen gets curated in there as a big reference database, and it's updated roughly monthly. It's been hard to keep that pace this year with coronavirus. And underneath that, we have a lead curator. So essentially this database, which looks like a website and is entirely human readable, also provides completely computer readable data. So you can write algorithms. And so there's a whole knowledge base around antimicrobial resistance, the genes, the mutations, the drugs, the mechanisms, their distribution among species, all this information. All that has to come from the scientific literature. And roughly there's about 4,000 papers published a year that we have to read. And so old school curation where you sit and you read them is simply not possible. So this was an undergraduate project. We actually build machine learning that reads papers for us and tells us out of the 500 papers that came out this month, we want you to read this one first because we think it has the highest body of information for the database. And don't read this one because there's nothing of value in it. You've already got all that information. So we've really moved into natural language processing and machine learning to really keep up with the scientific literature and keep knowledge bases accurate. But once you have all that, what do you do? So this is a picture of McMaster's neonatal intensive care unit. And we get, these are babies who are born premature or born with leukemia, or, these kids are in trouble from day one. And they often get infections, and those infections are often multi-drug resistance. You have to be as non-invasive as possible. So the most common thing we do is we take a rectal swab, right? And we sequence that fecal material. But most of the material is the patient itself, the human. And then the rest of it's all the bacteria that are supposed to be there. And the tiny little needle in the haystack is the gene and the pathogen that's causing the problem. So just like Dr. Schellhorn said, we take all of our computerized knowledge and we leveraged it to make a lab assay. A tool that would capture just the DNA we want from that fecal swab. So we can just sequence the stuff we were interested in. And so we did this with Jerry Wright's lab, these bait capture methods, and you still get fragmentary, millions of data points, really messy data. So Amos here, a developer, built a bioinformatics pipeline, and an undergrad built this point at the end called Kamers, which allows us to take this messy, fragmented, partial data. And in this case, in these patients, last year we predicted it was a Kleb pneumonia infection, and it had a whole battery of drug resistance genes, many drug resistance class. We still don't know how it got into the NICU. It possibly from a parent, probably through vaginal delivery. But at least now we knew what we were dealing with, and we could go to the clinicians, and there were still drug classes left, so we could treat those kids. I'll point out this last step, this Kamers, to say, fine, we found the AMR genes, but who's the culprit? This is essentially another term for what Dr. Schellhorn was talking about of these signature sequences methods. This type of bioinformatics is getting extremely common for identifying pathogens, and we were inspired by Dr. Gupta's work. But, once you do that, you get that DNA sequence from your pathogen. It's really just a list of genes and mutations. Right? And we have a lot of knowledge in our knowledge base, but just because it has a gene doesn't mean a bacteria is going to use it. And so trying to predict what a bacteria will do in a real living infection is hard. So this figure is trying to predict from sequencing hundreds upon hundreds of E. coli. This data says actually 115, but we're bigger now. And trying to predict from all these clinical strains from current existing bioinformatics, whether we can predict the drug resistance. The take home is if you see orange or yellow, that's failure. So you can look at the nitrofurantoin, right? We failed in almost every case. We totally got the wrong prediction of what the bacteria was going to do in the infection. And this is what I call my generation's bioinformatics. But when Cara, our PhD student, instead applied machine learning to this approach, it ends up looking like this. And so this is really where the field is going to go. We're going to have massive data sets generated by traditional bioinformaticians and biocuration, but from that, we're going to start training machine learning and artificial intelligence. My dream scenario, which is one we're working for, is the Canadian North with tuberculosis. Right now, if a kid comes in coughing poorly, a nurse practitioner may fly in every 2 weeks, take a swab, take it to Ottawa. Tuberculosis can take up to 6 weeks to grow on a plate, another 3 to test. So by the time you find out there's a new drug resistant strain, half the community has it, months have gone by. I want to have a day where that nurse practitioner flies in, asks the kid to cough in a cup, and they've got a miniature sequencer and a satellite phone with them. and it gets sequenced right then and there, and then AI immediately tells the Minister of Health to send a response team. And so to get rid of that time, that's the type of tools we're driving toward. It'll be more engineers in the end that do that endpoint, but we got to build the starting points. So just an idea what we do with drug resistance. We have this database, 3 years, 1.5 million page views. This map is every public health agency in the globe that used it in a 3-week period. It's highly cited, and now the data gets used. As I said, people can use it, but more importantly, other databases and other algorithms use it. So computers talking to computers. So we're informing other databases and other tools. And as I said, roughly every 5 minutes, a superbug gets analyzed by this sort of network of tools now. And it's very much an international collaboration. The biggest players are us, the Americans at the National Library of Medicine, the Danes and the French are the biggest working, and we all work really closely together, plus starting increasingly with companies. And the last bit, so we are a surveillance group, so no surprise, coronavirus came. We are part of the national effort to use genomics as a surveillance of coronaviruses with the Public Health Agencies of Canada. So we all have 2 jobs right now in the group. And in fact, my team was part of the one that isolated the live virus in Canada. So we did all the genomic verification as it was cultured and grown to prove it really was SARS-CoV-2 and it wasn't some weird mutated form. That virus now has been shipped to over 100 companies and public health agencies for vaccine and drug development. So as a genomicist, bioinformatician, you can have a huge impact to be involved, especially when you collaborate with good lab colleagues. So we run this sort of part of a national, and we're responsible about 1/3 of the patients in Ontario. We sequence every nasal swab that tests positive, and we find the viruses and we look for mutations. And we use those mutations for the detective work, trying to figure out how it moves around, find out if an outbreak is really an outbreak, or is it just a chance people walking in the door with different strains at the same time. But we're also screening for mutations. We don't want to spend all this time and money building vaccines just to find out the virus is already mutated and the vaccine doesn't work. So we're always doing that. But say Ramsey in the middle there is one of my clinical colleagues. He handles probably half of Ontario's tests per day. Tests are based on PCR, and if the genome mutates at the PCR binding site, the test will fail and you'll get a false negative. So we're constantly doing that surveillance for that. And then on the right, Aaron G. Banerjee, they are the biosafety 3 level culture group in Ontario. So they do all the experimental work. And when you grow a virus in culture, you got to make sure it's not evolving to adapt to the culture, that it actually still is the same virus that's in people. So we're constantly sequencing for them to make sure that their lab experiments haven't drifted off in some weird space as the virus mutates to adapt for cell culture. And so far, so good, to be perfectly honest. And then our biggest contribution, a group of graduate students and people around Canada built what's called the Signal Pipeline. This is a standardized bioinformatic workflow for working with this virus. And this pipeline now is the only pipeline that's used by the Public Health Agency of Canada. And at this point, 15% of the Canadian data has run through this, through my shop and doing that. And so this was tough. We actually, the guy there from the perimeter is a black hole physicist, but probably one of Canada's best programmers. And he volunteered his time for 3 months. A really multidisciplinary crew. We banged this thing out in 6 weeks. There wasn't a lot of sleep for 6 weeks, and all the standards with a new virus we knew nothing about. And so this was particularly Jalise on the top left. There's a PhD student who led this, just extraordinary work. And then it's Finlay McGuire. He's a machine learning specialist at Dalhousie. So this is the Canadian data. So if you look along in time from when the virus came to where it is, these are all the strains in the globe, a snapshot. All the red ones are Canadian. So when the virus got into Canada, we basically got every single lineage because the airports were open, people were coming. And then we did the shutdown. We asked people to stay at home. And most of those lineages went extinct in Canada, and only a few survived. And so the genetic diversity of the virus is lower in Canada, which is really critical. If you have high genetic diversity, it may recombine to become vaccine resistant. So what's happening right now, the second wave, we're starting to worry. Lineage numbers and diversity is starting to go up, and then we need the population to suppress it, never mind to save our beds in our ICUs, but we got to slam the genetic diversity of this thing back down again so it stays the same virus and doesn't evade vaccines. So this is genetic evidence of how good that shutdown went and how good people were eliminating the virus. So just thank you slide. You know, this is, I'm not going to mention my national and international collaborators. There's about 100 of them. It's endless. But again, a lot of this was engineered and run by undergraduates under the supervision of graduate students. In fact, Emily here, who's on the middle left, is in the 4th year of the BDC program. She's personally handled about 15% of Canada's genomic surveillance on that. And when she joined me in May, she didn't know how to program or do command line or anything. And so we trained her up, and now she's a national leader. And there's a few people on the call that are thesis students in my lab who come January, they're going to get their workups too. So it's a really good thing. I will admit, you know, that question about training, McMaster's shop, when I got here, was really thin on bioinformatics. But like both the speakers said, sequencing these tools are becoming cheaper and faster. So many labs that you might not think are bioinformatics labs actually will have a bioinformatics wing. So in biology, Dr. Wilson's lab, Jerry Wright's lab over in biochemistry, Mike Surratt's over in medicine. These are molecular biologists, chemists, you know, biologists, but they're using genomics and bioinformatics as well. And so even though I'm a completely dry lab, like say Dr. Golding in biology, and we're strict, you know, informaticians, the diversity of bioinformatics being done on this campus is becoming remarkable because there's a revolution when it comes to DNA sequencing. And there we go. Thanks so much. And happy to answer any questions.
I did have a question. It's not really, maybe it's relevant, I don't know. But you mentioned that you work with a lot of international partners and stuff like that. And obviously, like if there's an outbreak, it's going to be somewhere that's maybe less developed or et cetera. So how do you sort of collect that data from, you know, overseas or wherever?
You bet. You know, that's the world that we want to build. It's far from perfect right now. Part is that the people globally that work on infectious disease at the genomic level, bioinformaticians do it, is a pretty small community. We all kind of know each other. And because pathogens travel, we have no choice. You know, I have to pay attention to what happens in New Zealand or India or Kenya, for example. So over time, we get to know each other. There's international training events. The data sharing. So we build a lot of, I didn't really talk about the standards of how you share data and how you organize data. I would say 15% of the world has adopted it at this point. There's big drives right now because of coronavirus, because those gaps really hurt. And so I think it's going to improve. How you do that, it's a combination of things. You publish, right? So people read your work and you define the standards and they say, wow, that's valuable, I'll use it. But you partner with public health agencies around the world and you prove it. You prove that it works. You prove that it helps. The commercial side is huge. Having 30 or 40 companies involved in this or licensing the rest, when companies, you know, make profit off of it and it works, that proves that it's highly valuable. People adopt those standards. But it's tough, right? Public health agencies are not rich. And not all of them can DNA sequence, and certainly not all of them can afford a bioinformatician. And so you'll see countries like Canada and the US and a few others that basically give free service. And that, so no public health agency pays a cent to me. And in fact, if like Singapore called last week with cases and we just volunteer our time helping them. But yeah, it's got a long way to go. Informatics as a whole, data sharing is getting better. It's better, like in the first speaker around, you know, genome-wide association and gene enrichment. There's lots of great standards in sharing. Infectious disease is about 10 years behind, but it's coming. But actually, I've spent the last 8 years being an evangelist, right? Hitting the road, talking to people, convincing them and helping them to see it adopted.
Thank you.
You're welcome. Thanks, Tina.
We have a couple of questions in the chat. So Krishna is asking, what coding language do you teach your lab students? Yeah,
so the vast majority of students that come into my lab have no experience whatsoever. They can do their email and PowerPoint and YouTube. We very rarely take engineers on that. And occasionally there might be like a biology student that's self-taught. So the students are in my lab and are about to start in January, about to find out it's not the most pleasant of experience. But as a starter language, I always refer to Python. You know, my generation, we used Perl, and I still do a lot of Perl because I'm fast and furious at it. But Python, and there's a lot of great Coursera training and all the rest. We do that, and most students do a degree of that in our lab. Not everyone becomes a programmer in my lab. There's a lot of bioinformatics tools that you can just use. You don't need to know how to program. So you're really just learning how to do high performance computing. But generally, as the students work on the problems, sooner or later, they identify a gap that needs to be filled, and they start learning how to write software. We train it because we have a couple real hardcore engineers in the lab, and you learn by doing. But I highly recommend Python as your starting point. We probably use 40 different languages. Python is the workhorse that does the bulk of the work. All the other 39 are very special needs, like web presentation, or like we'll go to Fortran or C when we want to get every little thing we can out of that chip to make it fast for the big data sets.
A second question in the chat says, what kind of genomic tools do you typically use in the lab? And would analyses used in human genomics be used for microorganism or model mammalian organism studies as well?
Yeah, so I'll answer the last part first. So it's really winding down in my lab, but for 15 years, we were very active in environmental toxicology, looking at pharmaceuticals, metals, contaminants in the environment. And often we'd use zebrafish as our model organism to expose them under controlled conditions and do transcriptomics and metabolomics. And so that's when we did our model work. We don't do a lot of that now. A lot of those experiments, they don't really need me anymore. There was that discovery phase of big high throughput genomics, and now they're down to working on the regulators and the fine details, which they don't need me for. For the surveillance, when we're dealing with pathogens, we do, when we want to do experiments, we have model E. coli strains on the rest. I'd say it's my sister labs. We're bioinformaticians. They don't let us touch pipetters. But often I'm dealing with some variant like Pseudomonas that I haven't seen before that shows up that is different. Sequencing technology is interesting. When I had my company, and especially when I was an American scientist, you had to have your own shop. Now it's very much, there are many sequencing facilities. So if I want data on, say, an outbreak at St. Joe's, I can coordinate with the clinicians there for them to take the samples, ship them to the sequencing core, and 2 days later, the data shows up at my lab. I don't actually touch a thing. And the sequencing technology used is really depending on your question. There's an increasing diversity. For a while there, everything was Illumina, but we just bought an MGI from China, really interesting device, high throughput. These miniaturized sequencers from Nanopore have turned out really, really important for complex infections. And so you just use what you can. So for AMR, we're often doing as much research as surveillance. We don't worry too much about the cost, and we pick a device that gets it done in reasonable time. For SARS, where you're trying to do live sequencing, then I've got tens of thousands of patients. I have to make it as cheap as possible and as fast as possible. And so now you pay real close attention to your sequencing technology. And so it really varies. And I will recommend, I say to every student in my lab, if you want to be a good bioinformatician, and many students in my lab don't, they want this as a skill to add to wherever they're going. But if you want to be a good bioinformatician, you better be at the bench for a while. The best bioinformaticians are ones that have done bench work and can understand the nature of that, or in the clinic and understanding the clinical problems. And so that includes DNA sequencing. I think every bioinformatician should have done some DNA sequencing. So Dr. Shellhorn mentioned Giardia lamblia. It's a water pathogen. So I was the first Canadian to do a large genome project. I sequenced that genome. So I spent years at the bench sequencing, and it made me a much better bioinformatician as a result.
I have a question. Hi, Dr. McArthur. Thank you for that. It was a really great presentation. Always learn something new. I was wondering if there are any professional societies or conferences that you'd recommend for someone who's interested in human genomics or even bioinformatics as like a whole.
Yeah, there's increasingly that. There's two, I guess three I've traditionally gone to. There is the International Society for Biocuration. So that's a specialty of how you build databases and how you build knowledge bases. It alternates Europe or the Americas each year. I have no idea where it is now, probably virtual. The other one I really like I advise joining is the International Society for Computational Biology, ISCB. They run an international conference called the ISMB. I can't remember what it stands for, and they partner with the European societies. This is a great conference. Again, it alternates Europe, Asia, or North America. Big mega conference. but it actually, like 3 or 4 days of just training workshops even before the conference begins. And then in my field, the American Society of Microbiology has specialist meetings. So there's one in December, which is the genomics and bioinformatics of infectious disease, which is a really influential one for that. That's often where I meet most of my colleagues. But I think as a starter point, the International Society for Computational Biology is probably your best choice. And then if you have a domain of biology you're interested, so if it's cancer you want to look for those societies as well. Increasingly, like if you're a member of the Cancer Research Society, those meetings will have a session that's heavily genomics and bioinformatics. It's unavoidable. Whereas it used to be very separate. But I go to the ISCB meetings once every 3 years, roughly, because I find if I don't go any more than that, I start to fall behind. That's where all the new stuff goes. But I love their training environment. The other big one I recommend to all of you, there are the Canadian Bioinformatics workshops. These will all be virtual this year. They cover huge range. There's one on infectious disease. There's a training workshop on R. There's one on metagenomics. There's one on transcriptomics, you name it. They're usually very affordable. And some of them, you got to be in a PhD program to get in. Other ones are for starters at the beginning. They're affordable, they're very good. And actually it's taught by profs, so you get to see the labs across the country that might interest you for future parts of your career.
Thank you so much.
All right.